Today we will be discussing DNA replication in prokaryotes. DNA replication engages a large number of proteins and enzymes, each of which plays a critical role during the process. This process is fast and occurs without many mistakes. Origins of replication consist of particular nucleotide sequences, and this is where replication begins. The origin of replication is recognized by certain proteins that bind to the site. An enzyme called helicase unwinds the DNA by breaking the hydrogen bonds between the nitrogenous base pairs. As the DNA opens up, replication forks are formed. Two replication forks are formed at the origin of replication, and these get extended bidirectionally as replication proceeds. Single-strand binding proteins coat the single strands of DNA near the replication fork to prevent the single-stranded DNA from winding back into a double helix. DNA tends to become more highly coiled ahead of the replication fork. Gyrase, which is a topomerase, breaks and reforms DNA's phosphate backbones ahead of the replication fork, thereby relieving the pressures that result from this supercoiling. RNA primase synthesizes an RNA primer that is about 5 to 10 nucleotides long and complementary to the DNA. DNA polymerase III uses this primer to synthesize the daughter DNA strand. On the leading strand, DNA is synthesized continuously, whereas on the lagging strand, DNA is synthesized in short stretches called Okazaki fragments. DNA polymerase I replaces the RNA primer with the DNA. DNA polymerase can now extend this RNA primer, adding nucleotides one by one that are complementary to the template strand. This requires a free 3' OH group to add nucleotides by forming a phosphodiester bond between the 3' OH end and the 5' phosphate of the next nucleotide. This essentially means that it cannot add nucleotides if a free 3' OH group is not available. RNA primers are now removed and the gaps are filled. DNA ligase seals the gaps between the Okazaki fragments, joining the fragments into a single DNA molecule. The 5' to 3' exonuclease removes nucleotides from the 5' end of the primer, so RNA primers could not be removed from the DNA during replication. The primers could not be replaced with DNA.